Today I will present some new and old methods of Fourier modulation microscopy, and in particular some novel methods involving dual and multiple illumination. In conventional dark field, the illuminating light passes outside of the objective. In the right side panel we can see an internal dark field method that can pass light into the objective and block the undiffracted light or zero order light at the objective back focal plane. This is a Fourier plane where patterns of diffracted light are formed. The axial method of George Piper uses a narrow axial beam that is blocked by a small disc stop. Bright field can be superimposed, for example, using a custom annulus mask and fine adjustment of the iris. The bright field intensity must be restricted for proper contrast. Another example is the single sideband edge enhancement method, or SSEE, using a semicircular illumination beam and a semicircular Fourier stop, or half stop. The left panel shows a pure dark field setup. The right panel shows a method to substitute a semi-opaque carrier attenuation filter to allow some direct light to pass. A variant design adjusts the bright field component by sliding a straight edge condenser mask to form a variable slit. We can use the term variable graded field for similar multimodal methods that blend bright field with internal dark field. We propose a novel dual illumination system that introduces a secondary beam to generate a pure dark field component, preferably in colour, which can be superimposed onto a primary bright field image. A strip stop or other mask shapes can block direct light reflected from a small adjustable mirror below the condenser. The so-called pencil beam can be aimed either to the optical axis or to other adjustable angles. The central or axial beam setting gives luminous appearance of internal detail, absence of typical bright edges seen in conventional dark field, and it gives deep field suitable for thick samples. In the peripheral or edge position, the resolution is doubled in one direction. The depth of field is relatively shallow, and the asymmetry generates a relief contrast. The superimposed bright field can be set up with asymmetric wide aperture if desired to achieve some relief contrast and to maximize resolution. Variants of Reinberg colour contrast can also be achieved. A slightly more sophisticated method can use a dichroic mirror system, such that the translating small mirror does not obstruct the primary beam. Another proposed method, which I have called dual illumination Schlieren microscopy, uses a modification of SSEE. The secondary beam is set off-axis to strike the half-stop only. The beam shown here has a relatively wide diameter, but in principle could also be made more narrow if desired, or as a slit beam. The half-stop systems can be adapted using a polarizing sideband attenuator system, using a rotatable circular polarizer and a fixed semicircular polarizer, both mounted below the stage to attenuate a semicircular sideband. The next figure shows a hybrid method that uses the strip-stop concept and its secondary illuminator to modify an SSEE microscope. Here, the dark field component is fully asymmetric. Next, we can see some schematics for single sideband microscopes. The right panel shows a 4F relay to project the Fourier plane to an easily accessible external location. The next diagram shows a folded version of a 4F conjugate relay. In this example, we show a strip stop mounted in the Fourier modulation plate. The plots here show modulation transfer function curves indicating that SSEE has superior high spatial frequency content compared to bright field, Zernicker phase contrast and DIC. Masking or modulating effects can be achieved using pixel-based liquid crystal spatial light modulators, or SLMs. These thin devices can be used both in the illumination and in the Fourier plane. SLMs can modulate intensity, phase, polarization, and colour for diverse methods. A method of Weigel et al. Uses a narrow axial dark field illumination beam coupled to an unusually wide axial Fourier stop that acts as a high-pass filter to block most of the low-frequency image components along with nearly all the diffuse scattered background light. Only a very low-intensity image is captured, suitable for detecting gold nanoparticles or single fluorescent molecules. However, the method achieved an extremely high signal-to-noise ratio. The above method could be emulated using a half-stop and the adjustable pencil beam. In the peripheral position, the rejection of background light is maximised, along with the filtering of low spatial frequencies. As the pencil angle is adjusted, there is a trade-off between background rejection versus image brightness, making this variable rejection internal dark field microscopy method suitable for general dark field imaging or transmitted fluorescence imaging. And in this case, there are no filters required. Next, we can see a condenser-free microscope using a circular array of LED sources that are aligned to an annulus phase ring to give a type of phase contrast. Other modes, such as bright field and dark field, are also possible. Here we show a simple bright field system using a full-width annulus LED array. Here are some proposed modifications for condenser-free microscopes that can superimpose internal dark field images. This requires a conjugate relay system. The layout in the right panel is intended to create images in red, green and blue that each have maximised directional resolution in unique directions that are separated by 120 degrees. This gives 3 sharp images undegraded by lower resolution from the other colours. These can be bright field or dark field or hybrid, depending on the positioning of the Fourier stops. In practice, the secondary illumination sources would need to be considerably brighter than the typical LEDs. This could be achieved by tightly focusing the LEDs instead of letting them spread out at 60 degrees. Alternatively, we could use focused fibre optic sources. The next diagram shows a similar layout for fully oblique tricolour bright field without any other sources. Greenberg developed MOBI systems with 3 or 4 white beams to achieve stereo microscopy in high NA microscopes, or to generate asymmetric contrast images by turning off one or more of the sources. Various combinations could be used for asymmetric contrast. The last 2 panels illustrate proposed Fourier modulation methods for internal dark field and for multimodal imaging in white or in contrasting colours. In another example, Zuo et al. Used a thin film transistor SLM for programmable RGB colour illumination, including as a colour wheel, viewed here through a phase telescope, and also some simpler Reinberg methods. This particular study did not use any SLMs in the Fourier plane, but in principle these could also be applied. They investigated bright field, dark field, annulus phase contrast, oblique contrast, and bright-dark multimodal. Here we can see buccal cells in iridescent dark field compared to iridescent annulus phase contrast, showing colour highlighted edges. Opposite sidebands have different RGB intensities, so the effect on colour is equivalent to partial sideband suppression. We propose adding Fourier modulation to emulate the previous tricolour methods, or to create iridescent internal dark field with improved visualization of internal detail and with deep field. Here are some examples that involve alternating left and right half masks, either as Fourier masks or as condenser masks. If the so-called left image is subtracted from the right image, amplitude detail is suppressed to give qualitative phase contrast. If left is added to right, phase detail is suppressed to give an amplitude image. If the subtracted image is divided by the summed image, a so-called quantitative phase gradient image is achieved. Half masks and other masks can be used at either the condenser or the Fourier plane for an equivalent effect via a principle of reciprocity. Methods such as single half masking can benefit from digital image enhancement, where contrast tonality is weak and or where white background dominates in the image. Back in the 1980s, Inoue used video enhancement of DIC and also video enhancement of crossed polar methods. He demonstrated imaging of isolated microtubules that were only 20 nanometers wide. The general issue there is not so much the limit of line width detection, but rather the practical contrast. The upper part of the image shows a single microtubule. The lower part shows an overlap of 2 microtubules, which cannot be resolved. The real width of around 20 nanometers is displayed here as an artifact more like 200 nanometers in width. In conclusion, Fourier modulation methods offer a wide variety of applications. We hope the proposals for novel, dual and multiple illumination systems will be well received. Thank you.